Okay, so first of all, thank you to the organization for putting together such a nice conference, and of course to our company to provide me with the opportunity to speak here today and to introduce our neuroscience platform. So, as I'm sure most of you are aware, and as it's already been discussed earlier and you're in the field, a majority of drugs fail in clinical trials, both for Alzheimer's disease as well as for Parkinson's disease. This is, of course, due to there's a lot of different reasons for this. One of those has already been nicely addressed this morning in a talk regarding the blood-brain barrier, which is an important barrier to overcome to get the drugs into the brain. And the brain is, of course, a very complex system. And that actually leads me to one of the other important reasons why these drugs fail, is that we still don't know exactly how the diseases, like what's the pathology, like how is it exactly progressing from the start to the pathology. So the underlying disease mechanisms are unknown, and that is also in part due to a lack of, or need for better disease models and preclinical models that can help us predict which drugs should be selected to move forward to clinical trials. And of course, a lot of efforts have been done in the field, and I don't want to take away from that, but I think there's still room for improvement. And that brings me to the topic of today's talk, which is the platforms that we have developed in-house and that we can apply to your research to help understand which drugs you can select to move forward in clinical studies or to perform drug screenings and so forth. So our company is Creative Biolabs. We're a preclinical CRO located on Long Island near New York. We're in Shirley. Originally, we started in the antibody discovery and development field, so we have very broad expertise in that. But we also branched into other fields of life sciences, including neuroscience, which is a more recent development. And in our neuroscience platform, we have many different state-of-the-art technologies available, including microelectrode platform, electrophysiology for a microelectrode platform, and also live cell imaging, as well as confocal and super resolution imaging as well. And we have applied these technologies both for Alzheimer's as well as Parkinson's to generate models that can lead to better understanding of disease and to basically screen for drugs. And so I will introduce in the next couple of slides a few case studies that are relevant for both Alzheimer's as well as Parkinson's. But to do so, I will first start with maybe a short introduction to what are the mechanisms underlying neurodegeneration. Because neurodegeneration in general can have many different causes. There's a lot of them are listed here. This is not an exclusive list, of course. And we also need to understand, of course, neurons are not the only cells in the brain, as has also been mentioned earlier in earlier talks today. We have neurons, we have astrocytes, we have microglia. All of these are, of course, affected by all these different components or all these different potential causes for neurodegeneration. So it's not a standalone system. And one of those cell types that has been getting a lot more traction lately, in the last decades or so, has been the microglia. They're no longer considered just the glue of the brain, but they're actually found to have many different important functions for contributing to neuronal survival and health. And one of those is production of pro-survival factors. But they can also be affected by both age and inflammation. And literature, and these are just a few examples from literature, but there's numerous papers out there that show that age as well as inflammation can impact microglia function. Important functions such as phagocytosis, which is important to clean up debris, and also other phenotypes can occur in these microglia, such as increased levels of lipid droplets. Now we decided, okay, let's try to figure out what's actually going on in vitro. Because if we actually use an in vitro system, what we expect is to see, compared to the in vivo model, if you treat them with an inflammatory stimulus, you would expect to see also a decrease in phagocytosis. However, looking through the literature, and I see one of my slides is missing here, sorry. In the literature, what we found that if you treat the cells with an inflammatory stimulus, actually what happens is an increase in phagocytosis, which is contradictory to what is happening in vivo. So what we decided to do was to optimize the system, and we use here different doses of LPS, and we do a very careful time course. Let's see if this works. And we use our lifestyle imaging system to evaluate levels of cell death first of all, because we want to make sure that we're not affecting the viability in culture. And in blue, you can see the cell death increasing after treatment with LPS. So we decided, okay, we need to stick to the very short time points, acute treatments with LPS. And doing so, we actually found that by modifying our paradigm, we are able to decrease indeed the phagocytosis after short treatment with LPS. And this is a phagocytosis assay, which is one of the other assays that we have optimized. We have different cell types here, but what you can see here, we have different systems that we can use for this assay. But in this cell type and in this assay here, the little, sorry, I can, okay. Sorry, that was too fast. Is there a way to go back one slide? Oh, yeah, okay. Yeah, so here in the video you can see, yeah, okay, it's not funny. Anyways, I think you see here the little dots are the beads that will be taken up by the cells, and then they start to fluoresce in red, and that's a way to follow phagocytosis. And so in addition to that, we also followed another phenotype, which is a lipid droplet accumulation. And after stimulation with LPS, again, we do see, we are able to actually find this phenotype that's occurring in vivo, but now also find it happening in vitro. And we used this assay to screen some test compounds, and we were able to identify this compound. We tested at 2 different concentrations, and we're able to decrease the level of lipid droplet accumulation after this LPS-induced stimulation. Now, this is a very easy model system. It's only one cell type. And of course, like neurons, as I mentioned in the beginning, they're not the only ones in the brain or microglia. And therefore, we established a multiculture system. It's actually a triculture model in which we use both neurons, but also astrocytes and microglia. So it's a triculture system that we optimized in-house with a more relevant physiological, relevant physiological levels of presence of these different cell types. And when we stimulate them, again with LPS, we can see that they are responding to LPS, and that we can prevent this, again, by adding an mTOR inhibitor called rapamycin. We follow this here by dot plot and then quantification of these parameters. Now, this is a primary cell system, and of course, these can sometimes be difficult to work with in terms of you only have wild types available or healthy, but not relevant to disease, for example. And for example, in Alzheimer's or in Parkinson's disease, a lot of mutations are occurring. And for that, we have also optimized a human iPSC-derived system, which contain mutations or can contain mutations. And here in this assay, we have validated the electrophysiological parameters for these cells. So here, this is a live cell electrophysiology. Is a microelectrode array, and you can see these cells firing in real time. And this allows us to quantify over 40 different parameters that are relevant for neuronal electrophysiology. And we found actually one of those parameters here, in this case, was the burst duration. We were able to correct that using the test compound. In addition to this iPSC-derived model, we also have brain organoids. So these are 3D organoids that are relevant, in this case, for Parkinson's disease, because we have both midbrain as well as striatal organoids available. And actually, by combining these, we formed assembloids, which can be used and applied to Parkinson's disease research. In addition, we also have an in vivo platform, but I won't go too much in detail there. We have rodents as well as NHP models available. So overall, we can do many different things, including compound screening using either immortalized cell lines, primary cells, primary tricultures, iPSCs, as well as organoids, evaluate morphology, characterize functional changes, do MOA studies, and also customize assays of interest. And with that, I would like to thank everyone for your attention, and a special shout out to the team at Creative Biolabs for their help with setting up these assays and all the support. Thank you.